Hi, my name is Jin, and it's my pleasure to be able to present my company NEXEL here today at AusBiotech and Invest 2020. I've been working at NEXEL as a researcher and alliance manager for the past 3 years. And our company was founded in 2012. We've been doing business really from 2015, but now we've really grown into a really big company with a lot of people and very enthusiastic researchers. And it's my pleasure to show you a little bit of that. So the core of NEXEL's technology is pluripotent stem cell technology. We base ourselves as a spin-off from Korea University, which is one of the best universities in Korea. And we are also the first Korean company to license iPSC technology from iPS Academia Japan. So I'm guessing a lot of you aren't too familiar with what pluripotent stem cells are. So here's a short explanation. When you have cells in an embryo, these cells are able to differentiate into all of the cells in every tissue of an adult mammalian or human body. We call this concept pluripotency. This is the cell's capacity to differentiate into any type of cell. This natural process is yet to be fully understood, but a lot of researchers around the world, us among them, believe that this is a source of immense potential and that we are able to take this technology and make something more. So here I will present to you our two main pipelines. So both of these are based off of pluripotent stem cells. And the first one is the development of biotherapeutics. The second one is manufacturing human-induced pluripotent stem cell-derived cell products. Now we base these on our research of differentiation, which is what I explained in the previous slide. And we first looked at the secretome. So these are proteins that are secreted by pluripotent stem cells. We discovered our first candidate, MP011, from this, and we have further developed it and are at the stage of IND filing. And we have also discovered other candidates from those. We've also developed our stem cell products to develop in vitro hepatocytes first, and then we've continued to go and develop 3 successfully commercialized products, and we also have a cardiac safety pipeline. We will further expand on these pipelines in the future, but we also have in our vision that we will combine these 2 pipelines and our expertises to grow exponentially and develop successful cell therapies. But to give you a little bit more detail on our current pipeline, here is our introductory slide to NP011. So as I mentioned before, we discovered the protein from the secretome. It had anti-fibrotic and anti-inflammatory properties. We attempted to engineer this into smaller versions in order to optimize production and its therapeutic properties. We were successful in finding a recombinant protein that was more optimized for fibrosis resolution, which was our first POC in the liver. We further developed this and we had successful POC for several indications, including myocardial infarction. I have some data here. So we first looked at the liver fibrosis model, which was induced over several weeks by treating TAA. You can see on the graphs on the left that the fibrosis area is drastically reduced upon treatment of NP011. We then looked at the myocardial infarction model, which is induced surgically. You can see that once only treated with PBS, the cardiac hypertrophy happens and there's a large fibrotic area. With NP011, a single treatment was enough to improve cardiac function and fibrosis resolution drastically. So in conclusion, we believe that NP011 has shown impressive anti-fibrotic properties, and we have clinical phase 1 for myocardial infarction planned in 2021. Our other pipeline are iPSC-derived cells. And as I mentioned before, these are cells which are produced for in vitro research. Now, a lot of people ask, then why are you doing this? And the reason is that the drug attrition rate is very low, or the attrition rate is high. And the main cause for this is cardiotoxicity. This can also affect drugs even after the drug reaches the market and can lead to drug withdrawal. This is because the old methods to assess cardiac safety pharmacology are outdated, and HIPSC-derived cardiomyocytes are touted as the next way to assess cardiac safety. This is also recognized by international bodies, and we are participating in the international guideline revision by the ICH. So just to explain a bit what we are selling actually is, if you look on the right, this is a picture of our cardiomyocytes, which are produced completely in vitro, in the lab. And on the left, we sell those cells in a frozen vial along with the cell culture media. What you can do with these cells is actually do ECG measurements in vitro. So on the right, you can see an ECG measurement, which most people are familiar with from movies. And on the left, you can see that we can do the exact same measurement with our CardioCyte S product. This will change how cardiac safety is going to be assessed in the future. So our business model is that we have our first candidate, MP011, which has multiple potential indications. We are actively searching for open innovation or licensing out opportunities. We are also focusing on growing our iPSC-derived cell sales via local partners. We have partners in the UK, Japan, and China, but are looking for partners also in Australia or the United States. And we have also started to operate as a service provider for testing cardiac safety. We have a pretty big team here at NEXEL. We are a total of 45 people, but most of us are researchers, and we have a high number of PhDs, 12, which makes us really research focused. For what we are looking for right now is, just to give you a bit of an introduction to our financials, we have successfully done a Series B financing round last year of around $10 million US. We are currently preparing for IPO in 2022, but we are looking for partners for strategic investment early next year, or for partners who would join us in our pre-IPO. Now, thank you for listening, and this was Jin, and it was my pleasure to be able to present to you today.